Introduction: the TCA cycle, tricarboxylic acid cycle, also known as the Krebs cycle or citric acid cycle, is a central metabolic pathway in cellular respiration. It plays a critical role in the oxidative metabolism of carbohydrates, fats, and proteins, providing energy to cells in the form of ATP, adenosine triphosphate. What is TCA cycle? The TCA cycle is a series of enzymatic reactions that occurs in the matrix of mitochondria in eukaryotic cells. It converts acetyl-CoA, derived from carbohydrates, fats, and proteins, into carbon dioxide, while reducing NAD and FAD to NADH and FADH₂, which are used in the electron transport chain, ETC, to produce ATP. Why is the TCA cycle important? Energy production. The TCA cycle is a key pathway for energy generation. The NADH and FADH₂ produced donate electrons to the ETC, which drives the synthesis of ATP, the energy currency of the cell. Biosynthesis precursors. The intermediates of the TCA cycle are used in the biosynthesis of amino acids, nucleotides, and other important biomolecules. Link to metabolism. It serves as a hub for integrating various metabolic pathways, including glycolysis, beta-oxidation of fatty acids, and amino acid catabolism. Examples of the TCA cycle in action: carbohydrate metabolism. Glucose is converted to pyruvate via glycolysis, which is then decarboxylated to acetyl-CoA. Acetyl-CoA enters the TCA cycle to generate energy. Fat metabolism. Fatty acids undergo beta-oxidation to produce acetyl-CoA, which enters the TCA cycle. Amino acid catabolism. Certain amino acids are deaminated and converted into TCA cycle intermediates, like alpha-ketoglutarate, succinyl-CoA, and fumarate. Regulation of TCA cycle. 1. Citrate synthase. Step 1. Regulation. Citrate synthase catalyzes the condensation of acetyl-CoA with oxaloacetate to form citrate. Inhibitors. High levels of ATP, NADH, and citrate, which acts as a feedback inhibitor, inhibit citrate synthase. These signals indicate that the cell has sufficient energy, thus slowing down the cycle. Activators. Increased levels of substrates, acetyl-CoA and oxaloacetate, activate citrate synthase. 2. Isocitrate dehydrogenase. Step 3. Regulation. This enzyme catalyzes the oxidative decarboxylation of isocitrate to alpha-ketoglutarate, producing NADH and releasing CO2. Inhibitors. High levels of ATP and NADH inhibit isocitrate dehydrogenase, signaling that the cell has sufficient energy. Activators. ADP and Ca2 activate isocitrate dehydrogenase. ADP indicates low energy status, while Ca2 is involved in signaling during muscle contraction and energy demand. 3. Alpha-ketoglutarate dehydrogenase. Step 4. Regulation. This enzyme converts alpha-ketoglutarate to succinyl-CoA, producing NADH and releasing CO2. Inhibitors. High levels of succinyl-CoA, NADH, and ATP inhibit this enzyme. This occurs through product feedback inhibition, preventing overproduction of succinyl-CoA. Activators. Calcium ions, Ca2, activate alpha-ketoglutarate dehydrogenase, particularly in response to increased cellular activity like muscle contraction. Key reactions and energy yield. 1. Acetyl-CoA citrate, enzyme, citrate synthase, no ATP/GTP is directly produced. 2. Citrate isocitrate, enzyme, aconitase, no energy yield. 3. Isocitrate alpha-ketoglutarate, enzyme, isocitrate dehydrogenase, NADH is produced, 1 molecule per cycle. 4. Alpha-ketoglutarate succinyl-CoA, enzyme, alpha-ketoglutarate dehydrogenase, NADH is produced. 5. Succinyl-CoA succinate, enzyme, succinyl-CoA synthetase, GTP/ATP is generated, substrate-level phosphorylation. 6. Succinate fumarate, enzyme, succinate dehydrogenase, FADH₂ is produced. 7. Fumarate malate, enzyme, fumarase, no energy yield. 8. Malate oxaloacetate, enzyme, malate dehydrogenase, NADH is produced. Energy yield per acetyl-CoA, 3 NADH, from reactions with isocitrate, alpha-ketoglutarate, and malate dehydrogenase 1 FADH₂, from succinate dehydrogenase 1 GTP/ATP, from succinyl-CoA synthetase ATP production. In total, the TCA cycle generates 3 NADH 7.5 ATP. Since 1 NADH is almost equal to 2.5 ATP, 1 FADH₂ 1.5 ATP, since 1 FADH₂ is almost equal to 1.5 ATP, 1 GTP 1 ATP, total, 10 ATP equivalents per acetyl-CoA, this energy yield is critical for cellular respiration, particularly in aerobic organisms. Thank you.